Hello, everyone. Welcome to today's live broadcast, Best Practices for cfDNA and FFPE Sample Prep to Ensure Successful Downstream Outcomes. I am Brenda Kelly-Kim of LabRoots, and I'll be your moderator for today's event. LabRoots.com is the leading scientific social networking site, and we are proud to bring you this interactive web seminar. For more information, please visit us at www.labroots.com. This webinar is sponsored by PerkinElmer. PerkinElmer is a global leader focused on improving the health and safety of people and the environment. Our dedicated team of 7,700 employees worldwide are passionate about providing customers with an unmatched experience as they help solve critical issues in human and environmental health. Our innovative market knowledge and expertise help customers gain greater insights into their science to better protect our environment, our food supply, and the health of our families. For more information, please visit www.perkinelmer.com. Here's how today's presentation works. We want to hear from you. Questions, comments, and even some answers can be submitted via the green Q&A button at the lower left of your screen. We'll try to get to everyone, but if not, we will make sure and follow up with you by email. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, please let us know by clicking on the support button at the top right or the Q&A button in the lower left. I'd now like to introduce today's speaker, Dr. Alex Lopez. Dr. Alex Lopez received his PhD in biology from Cleveland State University in 2002 with a concentration in molecular parasitology, and he collected his MBA from Cleveland State University in 2004. He received his Bachelor of Science from the Ohio State University in 1995. He joined Chemagen in 2010 and subsequently PerkinElmer in 2011, following the acquisition of Chemagen Technologies. Prior to that, he worked as a research associate for the USDA ARS at Cornell University and had a postdoctoral fellowship at Case Western Reserve University. I'll now turn it over to Dr. Lopez for his presentation.
Thank you very much, Brenda. Hello, everyone, and good morning. I'd like to present my talk on the best practice for circulating cell-free DNA sample prep, ensuring successful downstream outcomes. In this talk, I want to go over the main points of what I want to discuss. So what I'd like to do is just give a brief introduction to cfDNA, as well as following with workflow solutions of cfDNA, specifically focusing on pre-processing. And then the heart of this is the circulating cell-free DNA extraction solution presented by the chemagic automation technology. Afterward, we'll discuss post-extraction analyses, as well as a special research collaboration effort between PerkinElmer and Swift Biosciences in a poster we presented at ABRF recently. And this shows an example of the complete workflow. And then finally, I'd also like to talk about additional research applications for DNA extraction with formalin-fixed, paraffin-embedded samples. The reason the tie-in between circulating cell-free DNA as well as paraffin-embedded tissue is that both are very difficult to isolate nucleic acids. Quality is lower in terms of, as well as yield, so that there are certain challenges to isolating this. So I will start by presenting with the circulating cell-free DNA background. So what is cfDNA? So it's basically DNA that's circulating in the bloodstream. It basically can originate from the breakdown of cells, whether it's, white blood cells and so on. Also, you get some from breakdown of pathogen, as well as apoptosis and necrosis. And typically, they're a tool for the research of non-invasive prenatal testing, as well as transplant control and sports medicine. Pathogen detection in terms of an indirect effect of what pathogens do to the fragmenting of DNA in terms of being in this size area with circulating, as well as a cancer research in terms of what a lot of people call circulating tumor DNA or ctDNA. This is also commonly referred to as liquid biopsies in terms of being able to take the plasma, isolate these small fragments of circulating cell-free DNA, and being able to analyze and use this for any sort of research testing. So some facts about circulating cell-free DNA. Essentially, it's a needle in a haystack, or even more so, as my colleague likes to say, it's more of a piece of hay in a haystack because you're essentially isolating a very small piece of DNA out of a sea of other DNA, and the challenge is to really get a good, clean prep, not only just to isolate yield, but also to get a certain purity that will allow you to use this effectively and study effectively a number of downstream analysis such as next-gen sequencing and so on. So essentially, it represents a very small portion of the plasma. So basically, 150 nanograms of circulating cell-free DNA per mil of plasma. It includes all somatic and fetal DNA as well as wild type and mutant, and the target fraction is only 1 to 30%. And in terms of mutation frequency, especially with interest to ctDNA, it's only about 1 to 2%. So you're really, first of all, getting a very small portion of circulating cell-free DNA out. And then out of that, if you're going to do any sort of mutation frequency, it's even smaller. So to ensure reliable results, you really need to use as much plasma as possible. But I'll also talk about some of the benefits and drawbacks of using different amounts of plasma and how this would work. So essentially, this is a trace that shows, if you look at the trace from left to right, you have your lower molecular weight marker on the left, and on the extreme right, you have a higher molecular weight marker. Typically, circulating cell-free DNA comes at a fraction of about 168 base pairs, which represents the wraparound of DNA around the nucleosome plus spacer. You also have these other fractions, 368 and 552, representing 2 or 3 turns around the nucleosome. Also, you see a little bit at the right near the high molecular weight markers, a little bit of genomic DNA, which is basically the challenge of doing this, in that the idea is you really want to have a good signal-to-noise ratio that will allow you to do a good downstream study. It's not enough just to purify it, but to really get a clean preparation from that. So let's talk about the workflow and really what I want to focus on in this talk. So first, we talk about blood collection, and there's some aspects to how blood is collected. And then we talk about plasma separation, and then really what we're showing and really highlighting is the circulating cell-free DNA extraction using our PerkinElmer chemagen magnetic bead technology, which I'll go more in detail. And then finally, downstream analyses and results. So at first, you have the challenge of which tubes do you want to use for the collection. And typically, you have either regular blood tubes, which are standard lavender top EDTA citrate tubes. So both, and then you have this in comparison with specialized circulating cell-free DNA blood tubes, such as an example, STREC BCT cell-free DNA tubes. Both have benefits and drawbacks. With the regular blood tubes, they typically don't stabilize the white blood cells, meaning that if you're going to do any sort of circulating cell-free analysis, you have to produce the plasma very quickly, within 4 hours of collection, and thus they're not really suited for transport. One of the benefits is that they are definitely cheaper, and certainly there's, because these tubes are very popular and used for a lot of, whole blood extraction, genomic DNA, that they're well known to be compatible with a number of molecular tests. The specialized circulating cell-free DNA tubes, basically you get a minimal background of genomic DNA. So you basically stabilize white blood cells, and it also minimizes the DNA background, genomic DNA background. It's basically possible to ship them. They're glass tubes. They definitely cost more, but the other benefit is that they're well suited for storage at 3 to 5 days at room temperature. It's definitely possible. So, and let me go back to this last slide. One of the, I guess, drawbacks of using specialized cfDNA tubes, I think one of the primary ones have to do with cost, as well as procurement at different blood collection centers. One of the things, though, in terms of thinking about doing liquid biopsies, is that there's certain, drawbacks to using the regular blood tubes, because even though they're cheaper, you might also have a higher opportunity cost, because you have to deal with having a fast collection, plasma separation, transport, and being able to process them in a pretty short amount of time. So even though the tubes themselves are cheap, you can also have a lot of drawbacks in that you might have to do recollects, especially if the quality of the DNA is not good, either because it degrades or because what we see is a high genomic background, which I'll highlight in this next slide. If you look at the two comparative traces, the one on the left are regular blood tubes. So you could see at the left end, the low molecular weight marker. Let me just point this out. So right here you have your low molecular weight marker right here, whereas the peak of interest that we're looking at for circulating is right here, about 168 base pairs. What we see at the right end, though, is we see a lot of noise in the genomic DNA region. So essentially we have, with these EDTA tubes, it's essentially more genomic DNA background compared to the signal that we're trying to isolate. Whereas when we're using specialized tubes, again, such as the STRAC BCT tubes, you could see the 168 base pair peak represented by the arrow here, and you could see a much lower genomic DNA background, and then you have your high molecular weight peak here. So again, part of it is, availability of tubes, being able to use them, but there is definitely a benefit to using the specialized blood tubes in order to get the highest quality results. So that's one aspect. Another aspect is, how does plasma separate it? And what we've seen from different customers is different methods that were used. Essentially, people have used 10 ml starting blood volume, which can be common for, again, either stabilized blood tubes with a stabilizing solution, as well as EDTA tubes, and again, you have to process them less than 4 hours after blood draw. And you have to, so basically you have some mild centrifugation conditions in order to separate the plasma. So these are just examples of 3 customers using different centrifugation steps in order to collect. One of the challenges as well is you have the cryoprecipitate that you may see, especially in some non-stabilized blood tubes, which could cause some lipids to be found in the resulting DNA element, because this can present a challenge as well as other proteins. So what we mean by less is more, because even though earlier we were saying that we want to use as much plasma as possible, the goal is to really make sure that you get a clean isolation of the plasma from the separation. So the plasma essentially contains 4 to 5 mLs of, the blood draw contains 4 to 5 mLs of plasma, about 55% of the total blood. And then you have the buffy coat layer, which contains the leukocytes and platelets, as well as the 1% of total blood. And then below you have the erythrocyte layer. So the idea is that one has to be careful to isolate plasma that does not disrupt or interfere with especially the buffy coat layer, so that we don't get unwanted leukocytes, which then can contribute more genomic DNA to the background. This can also be achieved, so just some facts about what you find in the different fractions. So essentially with plasma you have water, proteins, electrolytes, circulating cell-free DNA, which contains all of various forms of circulating cell-free DNA. 60 to 100 million leukocytes or milligrams of DNA found in the buffy coat, and then you also have a lot of mRNA found in the 50 billion erythrocytes. So as I was saying, with the buffy coat, you really don't want to disturb this. So there are methods to either be very careful about removing the plasma, either through serological pipetting, or there's also automated measures using either lasers or cameras in order to isolate the plasma without disrupting the layer, using specialized liquid handling platforms. So what, again, as I established earlier, there are definitely challenges to isolating circulating cell-free DNA from the samples. So essentially, again, you're going to have, in many cases, more than 4 mL of plasma. You're going to have a low amount of target circulating cell-free DNA. You're going to have small fragments. And then you also have to deal with cfDNA protein complexes, as we noted that there are the DNA is linked with the nucleosome, so essentially you have these complexes, but they have to be broken. So in addition, in plasma, you also find a lot of high protein content as well, such as immunoglobulin that you find in these. So essentially you need to really get the highest recovery and also get a purity that really works with a number of sensitive downstream research applications. So what I want to discuss is how do we fit in terms of the chemagic solution using our patented magnetic particles for doing this isolation, and how can we essentially provide a solution that really gives you a nice pure product as well as getting a nice yield down for a number of downstream applications. So basically with our technology, it's a magnetic bead based. I'll go into the structure of the magnetic beads themselves, but essentially what makes us different are our magnetic beads themselves, as well as the method of how we use the automation technology by using a combination of magnetic separation through an electromagnet and also the ability of a stirring motor to turn the rods so that you can get an efficient mixing of particles when the magnet's off. So if you look, you have the electric coil, which basically the electric coil power is on, so you have the magnetism. And then when that happens, you could looking on the picture on the right, let me just point this out. You have, when the power is on, you can see, first of all, the magnet's off and the rod motor is on and it's mixing. And then at the same time, you have the magnet bead collecting at the bottom of the magnetic rod covered by a disposable sleeve. So when the metal rod is connected to a stirring motor, when the stirring motor is turned on and the magnetic field inducing power is turned off, the rods can spin around 3,000 RPM, enabling a smooth and very efficient resuspension of the attracted magnetic particles. Okay, so we can see that one of the big benefits of our automation technologies, we have a very flexible format system that can work with both low volume and high volume. So the most appropriate or standard form is the SBS format, and this could work in 96, 48, or 24 well plates. So one of the most common workflows that I'll talk about, especially in our example with Swift, is our 24 rod head solution that could use up to 4 mLs of plasma. So if you look, you could see how the chemagic rod head 24 works and how the rods are arranged in a way that fits with the plates. So let me just draw this arrow here, and then you can see how the rod arrangement then works with the plates there. As well as we also have a 96 rod head that could work with either 96 or 48 well plates. And both of these can be used as well, and I'll talk about the different solutions we have with circulating cell-free. This also is the format that we use for the fixed formalin paraffin embedded tissue that I'll talk about at the end of the talk. And we also have the ability for very large samples, up to 10 mLs of plasma. We can use the 12 rod head, which essentially uses reaction vessels that just consist of a single 50 ml Falcon conical tubes. So with our separation technology, we essentially have switchable magnets and rotating rods. It basically transfers magnetic beads, not liquid. So unlike a system where you have a single tube in which you have a fixed magnet at the bottom and you have liquid being pipetted in, mixed, and then you have to have liquid pipetted out, here we're actually moving the beads from plate to plate or tube to tube, depending on the format of the chemagic system you have. So essentially we get a very efficient resuspension of magnetic beads, resulting in a high binding efficiency, as well as very efficient collection of magnetic bead results in a high recovery of nucleic acids. Efficient suspension of magnetic beads results in a high washing efficiency, and technology allows the processing of higher volumes. One of our strengths as well is we can show that we get no cross-contamination between the samples by very careful mixing that our rod and stirring motor is able to do, and a number of methods within our isolation system to ensure that we don't get any cross-contamination. So what are some of the solutions that we have for circulating cell-free DNA extraction? So one of the most common ones that we've seen with opportunities, as well as talking to a number of customers, is this sort of medium volume DNA extraction from 4 mLs of plasma. Now again, everyone's going to have different targets for the amount of plasma they want to use. So a lot of times, circulating tumor DNA research can involve, as little as 1 mL of plasma up to 4 mLs. Sometimes if one's looking at doing fetal DNA analysis for research, that basically you could use higher volumes, 4 to 10 ml. So I'll talk about really one of the more common workflows we have is the 4 ml plasma. So essentially, we take 4 ml of plasma that's been separated and isolated, clean, and we try to make sure that it's clear of cryoprecipitates based on the centrifugation conditions listed earlier. We add 1.6 ml of lysis buffer as well as proteinase K because, as pointed out, there is a degree of a lot of proteins in the plasma samples as well as the cfDNA protein complexes. So we really need to do a rigorous proteinase digestion. So we do this at 60 degrees. Afterward, the lysates are put onto the instrument, and with the addition of binding buffer and the magnetic beads, we have a pretty fast binding time for binding the DNA to the beads. And this is really essentially going after very low molecular weight DNA, which would correspond to the 168 base pair region. So one thing I'd also like to point out too is that we've seen that, with the circulating cell-free DNA, we see a range of about 168 bases, but we also see that with some circulating tumor DNA that we have lower molecular weight as well, that corresponds around 145, 147 base pairs. So the idea is that our solution can really isolate all circulating DNA for a number of different purposes. So after the binding, we have 2 repetitions of 3 mL of wash buffer 3 and 2 repetitions of 3 mL wash buffer 4, followed by a drying step that we implement on the instrument, as well as the transfer to elution tubes, and we essentially elute in a very basic tris low EDTA buffer to stabilize the circulating cell-free DNA in solution. So pointed out earlier, the chemagic 360, which is our one of our featured instruments for doing this isolation, will use 24 magnetizable magnetic rods, and they're arranged to fit in the SBS format 24 deep well plate. So just a little bit of detail on our technology magnetic beads. So some of our benefits. So our beads are different because of several things. One, we have a high degree of magnetite, which allow our beads to very efficiently collect on our rods when magnetized. So that results in a very fast magnetic separation. The hydrophilic bead surface with non-measurable protein or background for proteins results in very pure nucleic acid isolation. Especially in contrast to silica particles, these beads can be eluted at room temperature. And our specific bead surface with a high affinity to nucleic acids results in high quantitative yields as well as high degree of purity. And it's a very scalable process, as I mentioned, with the different workflows. So our unique MPVA or polyvinyl alcohol-coated beads, you could see on the left, you could see the size of our beads are approximately 1 micron in diameter. And you can see the electron microscopic picture of the dried MPVA magnetic beads. So the different kits that we have, the CMG-1104 refers to the 4 ml plasma volume that we have that we could run on our high throughput systems, either the chemagic 360 or chemagic MSM1, which are shown below just to illustrate. This is the chemagic 360, and this is the chemagic MSM1. So both instruments use the same rod heads, and essentially they're just 2 different versions of the instrument, the MSM1 being in the field longer, the 360 being the newer instrument. We also have our second kit listed, the CMG-1090-A, is a kit that still uses the 24 rod head, but instead uses 2 plates to bind consecutive 4 ml solution plasma lysates in order to do a complete 8 ml isolation using this, gives a higher throughput. And lastly, we have the CMG-1110, which refers to the chemagic circulating cell-free DNA 10 ml kit. And that's the one that uses our 12 rod head and basically uses 50 ml conical tubes. So in a sense, once the plasma is separated and you can use these tubes for performing the lysis, it's a very rapid and simple procedure for putting on the instrument. So as I mentioned, these results are very scalable. So if you look at an example trace, you see, again, with the low molecular weight marker, high upper marker, you could see what we get from 10 mLs of plasma, 4 mLs of input plasma, and 1 mL of plasma. Just to scale, take a step back before I move on, is we also do have a solution for 1 mL plasma using our 96 rod head that essentially we can split the 1 mLs into 333 microliters and use 3 plates in concert. So basically it results in a high throughput, 96 samples at a time, 1 mL isolation, especially, again, popular for a lot of ctDNA, especially if people are doing a lot of endpoint targeted PCR, such as epidermal growth factor receptor tests. So again, this gives you the idea of what we get in terms of the scalability, and essentially we get a 2 to 5 nanograms of cfDNA fraction per mil of plasma. So one thing I'd like to talk about, and I'll definitely expand on this during the discussion on the poster, is that there are a number of different, I think, means to measure the quality and quantity of the circulating cell-free DNA. So some of the comments are that with circulating cell-free DNA, it's definitely more challenging to quantify without any sort of advanced analysis, because essentially what works well for a number of genomic DNA, such as genomic DNA from either tissue, blood, saliva, and so on, using simple UV measurements such as a nanodrop works fairly well in terms of giving a good approximation of yield as well as purity. Fluorometric that would analyze, for example, double-stranded DNA, or can also specifically analyze single-stranded RNA, is also very effective for those sample types. However, as I mentioned, that we're talking about isolating a needle in a haystack or a piece of hay in a haystack, and essentially what that means is that we're trying to look at DNA amongst DNA, and the only way we can do that in terms of really being able to quantify circulating cell-free DNA is to use a method that actually takes size into consideration, and these first two methods really do not do that. So in order to really accurately quantify and qualify the amount of circulating cell-free DNA in your isolation, you really need either the use of a trace, so fragment analysis. PerkinElmer has a lab chip GX Touch that is capable of doing this, as well as quantitative PCR, digital droplet PCR, and then what is done sort of downstream of that would be next-gen sequencing in order to really give a good qualification of what, how good the isolation is. So essentially, I'll be talking about especially the one method to isolate or analyze using qPCR. And as you've seen with the earlier traces, having a trace is really helpful in being able to actually depict the amount of circulating cell-free DNA in the fraction. And you can also look at the genomic DNA contamination in that as well. So a number of downstream research applications that we use with our system can be used for whole genome sequencing, amplicon sequencing, SNP-targeted sequencing, said qPCR arrays, SNP arrays, as well as Baxon beads, essentially analyses. So in these next set of slides, I want to talk about the research poster collaboration that we presented at ABRF recently. And this involves a complete workflow between not only the chemagen technologies, but PerkinElmer as a whole, and also a partner with Swift Biosciences in terms of showing this research application that can be very useful. So this just shows the poster that we had presented, showing this workflow, and what I'd like to do in the next slides is to expand on a lot of these figures and talk about, in terms of both the ability to quantify as well as using our DNA for the next-gen sequencing analysis of this, as well as feature how Swift provides a means to really generate libraries using very low, such challenging samples such as circulating cell-free DNA and FFPE paraffin-embedded formalin-fixed tissue. So, as I said, there are definitely challenges, and as I said earlier, again, piece of hay in a haystack. We want to be able to show sequencing more from less input. So as you could see, when you're dealing with circulating cell-free DNA, you're dealing with a low quantity as well as integrity. So you can see high molecular weight DNA, and you get a high degree of integrity. It's a little easier to isolate. And then you have, if you're going from top to bottom, let me just show you the arrow right here, so you can see as you're going down, you have partially degraded DNA as well as whole genome array, paraffin embedded, and then circulating cell-free. So that is definitely the challenge. You're dealing with a lower amount of integrity, as well as, if you scale this with the quantity, a lower amount of quantity. So it is important to have methods that could really generate libraries, especially post-extraction, that can give you a good analysis of what the consists in the DNA and the information that you want to get from that. So I mentioned different means of quantifying circulating cell-free DNA. What I would like to spend a few minutes on is talk about a specific method that was used to calculate not only the yield of cell-free DNA using a quantitative PCR method, but also to determine the quality score that would be similarly represented from a trace. So what I'd like to, what this method is basically a method that uses the ALU repeats, which are essentially 300 nucleotide long, very abundant sequences in the genome, constitutes about 10% of the genome, and especially abundant in blood. And this method that was developed by T.B. Howe et al. In 2014 British Journal of Cancer article or manuscript that uses essentially two sets of primers that amplify 115 base pair amplicon and a 247 base pair amplicon using these allogenomic repeats. So what they did was they generated a 115 base pair amplicon that represents the circulating cell-free DNA, whereas the 247 base pair amplicon represents a genomic DNA background. So by doing this, you could look at the, when you look at the final concentration of genomic DNA, which is found in this column here, that is derived from the 115 base pair amplicon. So essentially you get the concentration, and thus you can calculate the yield. The quality score then is a ratio of the amount of amplification from 247 divided by 115 amplicon. So what they had done in their research study was come up with these different quality scores that, with regard to the ratio, that basically determine what they saw in terms of what constituted a good circulating cell-free DNA prep, genomic DNA, as well as what they found with a colorectal cancer patient. So essentially, they found that with a mean of 0.038, with a range of 0.029 or 0.29 to 0.49, they found that a good quality circulating cell-free was found in this region. They found from a colorectal cancer patient about 0.62 in their research study, with a range of 0.51 to 0.65. Whereas genomic DNA, an example of contaminated genomic DNA would have a score of 1. So as you could see from the study that we had 10 samples that we isolated using the chemagic 360 using the 4ml protocol. We essentially got scores in the range of 0.3 to 0.5, which would indicate a good quality of circulating cell-free DNA. As one thing just wanted to mention also is that this information is available as a technical note found on the Swift Bio website. So if you want to get more details, it outlines the primers, PCR conditions, and method that was used to determine this. So this also provides a nice way to not only quantify but also qualify circulating cell-free DNA. So as again, as I said, there's a challenge for working with difficult samples. So now that we've been able to quantify the circulating cell-free DNA, this goes into what Swift has in terms of their special library adapters that are able to work with difficult samples, such as FFPEs, circulating cell-free DNA, and so on. You could also have, some critical factors include DNA damage, size selection, as well as the depth of sequencing, how many rounds of sequencing you want to get for the genome. So basically, 1 nanogram of genomic DNA is equivalent to 167 copies of the diploid human genome, 334 chromosomal copies of any locus. and achieving 101% detection of a heterozygous mutation is equivalent to 3 chromosomal copies. So again, very challenging procedure for doing this type of analysis. So what Swift has are the what they call the Accel-NGS 2S+, and essentially they are special linkers that in method that allows for the use of damaged DNA as well as, again, challenging DNA circulating cell-free FFPE. So essentially, they're doing a dephosphorylation of both the 3' and 5' termini of the DNA fragment, which helps reduce chimera formation and improves ligation. And then they do a repair of both of the 3' and 5' ends, eliminating any damaged bases. They then do a subsequential ligation of the P7 and P5 adapters, and that also prevents adapter dimer formation and eliminates the need for adapter titration. Then there is an optional PCR step, but one of the key things is that this can be done PCR-free with low amounts of input, but is also, you can have the option of doing PCR, so you can see the different ranges of PCR-free inputs down to 10 nanograms with PCR inputs down to 10 picograms. And then you have the equivalent adapter ligation efficiency at all input levels. So using our DNA extracted from the chemagic 360, they're able to do with 2 of these examples. They were able to get with example 1 and example 2, 238 million and 254 million reads respectively. And you could see that they had a good average genome coverage of 14.6 times and 15.6 times respectively. And then you could see with the size distribution, after, with the circulating DNA, that you get a nice median insert size of about 169 bases. Essentially, then libraries were then sequenced on a HiSeq, Agilent HiSeq 2500. And this just shows a comparison of the genomic material to the circulating cell-free DNA. Again, the idea of either having using 100 nanograms of PCR-free versus 10 nanograms of PCR, you can get very similar yields of the circulating cell-free products we'd like to get for setting up sequencing. So what really is, this ends up leading to is that we have, with this workflow, we're able to show a detection of 1% mutation in cfDNA using hybridization enrichment technologies. So again, with this DNA that was isolated from the form alzoplasma on the chemagic 360, each sample represents an individual with unique ethnic background and detect mutations that only 1% of cfDNA was spiked in. And then with that, you can see how well with the different chromosomes and the different positions, chromosomes 2, chromosomes 12, you could see the effectiveness of the being able to detect this spike in sample, 1% A into 10 nanograms of B and 1% A into 10 nanograms of C. So it really shows that with this example, research example, that we could show a good recovery of the allele frequency, being able to simulate what can be done in terms of the capabilities of this platform. So yeah, they were used. These libraries are made using 3 samples and using the Accel-NGS 2S library kit, as well as the IDT Gen Pan Cancer panel as well, and then they were sequenced on the Agilent HiSeq 2500. So with that, then, that concludes a portion of that I wanted to talk about in terms of cell-free DNA. I would like to expand on some additional research applications. So again, since we talked about difficult to isolate samples, paraffin-embedded tissue, and again, you're dealing with lower quantity as well as lower quality, I wanted to show some of this extraction solutions that we have for these sample types. So essentially, paraffin-embedded tissue is estimated that there's 1 billion of these samples worldwide, and they're a common method for research and solid tissue samples. They're an excellent resource for genomic insights. They have a high throughput demand screening, and we also have a research collaboration with the Covaris truXTRAC system, which I'll also talk about, as well as a PKI workflow. So with the research product updates, we have a couple of examples I wanted to give. So we have our high throughput system using the chemagic MSM1 or 360, and essentially our kit number 1099 shows the isolated automated isolation of DNA from 5 to 15 micron section using an equivalent of FFPE tissue with a technology using the 96 rod head. So as I pointed out, the 96 rod head is what we use for the isolation of paraffin-emitted tissue. We also have a smaller instrument that's capable of isolating similar volume samples to our 96 rod head called the chemagic Torpedo, which can do 12 samples at a time. So this just gives an example of how we do. So compared with competitor A and B in the blue, our solution in green shows that we get a higher yield of total nanograms of samples in various different tissues, including breast, colon, liver, and lung tissues. So you can see with these different tissue blocks, we have shown to have a very good method of isolating genomic DNA from the FFPE samples. Additionally, with these two isolation kits, again, what we have also shown in terms of not only yield but also with comparison to competitor, we show a higher fragment size when we do a head-to-head comparison. So you could see with the different sample sizes, if you look on the left column versus the right column, these are our isolations. These are competitor isolations, so we can show a higher fragment size with these various different tissue samples with our platform using either the chemagic MSM1 360 or the chemagic Torpedo. We also have a research collaboration with Covaris, which uses a sonication or AFA acoustic focusing for removal of the paraffin-embedded tissue, and essentially it was paired up with our chemistry. We have a kit that will work for this solution as well. So if people have a solution that is using the Covaris system, we have a specific kit that works for us, again, using either the MSM1 or 360 with the truXTRAC DNA kit or with the smaller instrument, the Torpedo. So again, with a chief competitor, green versus blue, we show that we have higher yield of DNA from prostate, colon, and lung in comparison with a primary competitor. In addition, again, showing an average fragment size with either of our formats, we show that we have a very good average fragment size from prostate, colon, and lung as well. The thing that you see is a little bit of smaller size with the acoustic focusing, but it also depends on what you're going to be doing downstream with this DNA. So again, we just want to show that we're very adaptable. We have, with regard to paraffin-embedded tissue, we could work with a number of different means of removing the paraffin, whether it's either done through detergent, xylene, for example, using the Covaris system, that we have a solution for all these different types of deparaffinization. So let's summarize. So circulating cell-free DNA is typically observed as approximately 168 base pair peak. Again, the 168 base pair peak involves the wrapper on the nucleosome plus some linker DNA. And again, we see a slightly smaller size for ctDNA. Specific stabilized blood tubes for circulating cell-free DNA, such as the BCT or STRAC BCT tubes, allow for greater stability of cfDNA, but also have less genomic DNA contamination. Our automated system basically shows that we can have a very flexible workflow from 1 to 10 mLs of plasma, so different formats for different needs. Depending on what you're doing downstream, we have the solution that would be, offer the most economy of scale in order to give you the best, either the highest throughput for what you need, and also we can do a very higher amount of plasma in case you need to isolate a greater amount of circulating cell-free DNA. Qualitative analyses such as fragment analysis and qPCR show the specific yield of the targeted size of the circulating cell-free DNA and also represent and show the genomic DNA contamination. And through our research effort collaboration, we're able to show an entire workflow for the analysis of cfDNA from 4 mLs of plasma. And then as an additional research application of interest, we have a solution that works with a number of paraffin-embedded tissues, working with the Covaris truXTRAC workstation, as well as samples you may have by using traditional means of isolating or deparaffinizing samples. Before we get to questions, I just want to just give a couple of reminders. If you would like to learn more about our technology, please visit our booth. We have a number of reps that are in the booth to answer your questions. We also have a video showing the chemagic 360 workstation in action, so you can see in illustrated detail how the isolation works. And I'll also be available myself in the next hour from 11:30 to 12:30 Eastern Time to answer any additional questions if they don't get addressed in this question and answer section. And with that, I thank you very much for your time in participation in this tutorial.
Thank you, Dr. Lopez. Excellent presentation, lots of good information. Before we get to questions from our audience members, I just want to remind them how to reach us today. Questions can be submitted by the green Q&A button at the lower left of your screen. And we do have one right now in our queue. An attendee asks, does the type of heparin tubes for blood storage prior to plasma isolation impact the yield of cell-free DNA?
Thank you for the question. So with regard to heparin, and this falls under, not only circulating cell-free DNA, but genomic DNA. Heparin chelates with DNA, as being a anticoagulant, so that it won't affect the yield of the DNA isolation, but it would affect the ability, especially of doing any sort of PCR analysis, unless you use a specific kit to remove the heparin from the DNA. The isolation process or any extraction process would not remove the heparin from that. So no, it wouldn't affect the yield, but yes, it would affect the quality of downstream application depending on what you want to do.
Great, thank you. I do again want to let our audience know that Dr. Lopez will be taking your questions. If you just click on the green Q&A button at the lower left of your screen, type your question into the box that appears, and Dr. Lopez will be happy to answer your questions here today. Dr. Lopez, while we wait for any questions to come in, do you have any other sort of summary comments or advice for our audience members?
Thank you, Brenda. Yes, so if you have any specific questions in terms of your workflow solutions, what you can see in terms of either seeing what our system can do for you, and also even advice of isolating or analyzing circulating DNA, please feel free to reach out and ask us any questions. We've worked with a number of opportunities, and we realize, especially in the circulating cell-free DNA area, a lot of methods are very proprietary. There is some degree of customization with regard to that, but I think what we're trying to present is a workflow solution that really shows a nice workflow from all the way from sample collection to analysis that we can work with you, and showing as an example of how our technology plugs into a complete workflow is something that really shows how effective our extraction technology is, and what essentially what we can really help you with in terms of furthering your ability as a organization to better analyze circulating cell-free DNA.
Great, thank you for that information. We do have another question coming in. Will the PDF copy of this presentation be available?
Hi, thank you. Basically, essentially we can provide something to you. It just maybe collect some information, or just at least talk with our regulatory to make sure everything looks fine. But I think, we'd definitely be able to provide that information for you.
Great. Thanks. That'd be a great resource, I'm sure. I don't see any more questions in our queue, so I'm going to once again thank Dr. Alex Lopez for being here today and bringing us this information. I'd also like to thank our sponsor, PerkinElmer, and we'll leave it to Dr. Lopez to wrap up for us.
Thank you. Thank you very much again for giving me the opportunity to present this technology. One thing too, if you have, again, time to visit us in the booth or talk, we'd like to just from our information as well, like to find out some aspects of what types of analysis you're doing in terms of your cfDNA workflow. An example, how are you analyzing the extraction yield quality, as well as the type of blood tubes and also volumes, so that in addition to learning about what you're doing, there is the ability to, again, really see if we can gear a solution that we have with our flexible system to work well for your platform, or for your, at least if you're not doing currently, looking to do in the near future.
Great, and I did misspeak. We do actually have a question. An attendee asks, do you find there is any difference when you test ctDNA in different cancer types?
Yeah, thank you for the question. So right now, as we're kind of focusing on this from a research application, I think to that specific question, we have not explored that at this time. I think in terms of working with research collaboration partners, that would certainly be of interest. But currently, we have not, don't have any other data other than saying that that we do see, and this has been published in the literature, that the circulating tumor DNA does show a smaller size than the, let's say, circulating cell-free DNA from quote-unquote normal patients.
Great, thank you. Lots of good information here. I do want to thank our sponsor, PerkinElmer, for making this webcast possible. And again, Dr. Lopez for bringing the information to us and taking time with us today. And another question. Dr. Lopez, from start to finish for the workflow, how long does the instrument take to analyze the specimens? How long do you estimate it takes to prepare specimens for the chemagen, for cfDNA, different times, for FFPE? FFPE.
So I think from the question, so let me what I can do is I can kind of cycle back to the question about, or showing the workflow. Go back a little bit. So with the well, I'll start with FFPE. With FFPE, essentially what the biggest challenge is to basically deparaffinize the sample. A lot of times, and it depends on if somebody has already an established workflow with a manual procedure, there's a number of ways to go after it. Again, with the Covaris truXTRAC system, it's a very rapid, using acoustic adaptive focusing to remove the paraffin from that. It's fairly rapid. However, there's also, if one's going to use any sort of like a detergent or a xylene, extraction, that takes. I think what most people do is, especially with regard to the proteinase K digestion, they first need to do the removal of the paraffin, and then followed by a proteinase K digestion at 60 degrees. And a lot of times people are doing this overnight prior to loading that lysate on the instrument. Once that lysate is then generated, the runtime of the instrument is approximately 45-50 minutes. And that works for 1 to 96 samples. Using the high throughput system, the MSMR 360, as well, and then the low throughput system, the chemagic Pepito 1 to 12 samples. With the circulating DNA, that, in terms of the upfront steps, that all depends on, in terms of your blood collection when you separate the plasma and generating that plasma. Essentially, if we start with 4 ml plasma, We're going to incubate about, usually in most cases, 60 minutes at 60 degrees with doing a proper proteinase K digestion with a lysis buffer. And then the binding is 40 minutes, and then the drying 5 minutes. So essentially, you're talking about 2 hours for the extraction of circulating cell-free DNA from that point. And then what the 360 will do is produce the circulating cell-free DNA, and then in terms of downstream analysis, that's up to you as far as how one wants to quantify, either using a instrument that does trace analysis looking at size fractionation, or using a either quantitative PCR, digital droplet PCR for that analysis, depending on how long the thermocycling takes for that.
Thanks again so much. I also do want to thank our sponsor, PerkinElmer, to make today's webcast possible. This webcast is going to be available on demand through November of 2016, and you'll be receiving an email from LabRoots alerting you when this presentation is available for on-demand replay. We would invite you to forward that email to your colleagues who may have missed today's presentation. That's all we have for you today in this presentation. Thank you again to Dr. Lopez and PerkinElmer for bringing this information to you. We hope we see you again.